Professor Rieckmann is a leading German neurologist with over 25 years of experience in Germany, Canada, and the Gulf region. Of course, he is visiting Russell Hospital regularly over the last 10 years. He has an excellent track record both treating patients with neurological disease and outstanding clinician scientist. Professor Rieckmann will be updating us on pathology in multiple sclerosis. Professor Rieckmann, please.
Assalamu alaikum. Good morning, everyone. And thank you for allowing me to give the first presentation of today's symposium. These are really exciting times in MS research, particularly as things seem to converge. What we have learned from pathology, we now can turn into almost in vivo pathology, and is linked also to the pathophysiology of multiple sclerosis. Therefore, I slightly modified the talk in the title, as you can see it here. My disclosures, and on the next slide. So if we talk about MS pathophysiology as a continuum over decades of life, the continuum of a disease is more important than what we have been told in the past to split it in relapsing and progressive types of MS. Because we know that there is both aspects, almost always present in any case. And that depends on the time frame of the evolution of the disease. And it's our task, basically, to define which one are the most prevalent one. And thus, we can interfere best in the early phase of the disease. We have always taken into consideration that something is going on under the surface, already evolving into what we can see later on as clinical progressive disease. We have new parameters like RAW and PIRA, which try to define this based on the clinical appearance. And we know that even progression in most of the cases is associated with aspects related to inflammation. What we have seen over the years, going back to the very first description of pathology, that the overall hallmark of multiple sclerosis are the central vein accumulation of lymphocytes, and also the aspects related to the demyelination occurring in the white matter, but more and more also being described in the gray matter. And as you can see here on the right side, it is shown in dynamic evolution on advanced MRI technology, how that's going to happen. And I will deal with that a little later. So the classical immunohistology, as it is outlined here, where we, on the one hand, can define the chronic active lesion, and then the expanding lesion, and then the shadow plaques, something which is important if we try to turn the histopathology as we see it under regular condition on our slides under the microscope to in vivo pathology, is what we can see here, the accumulation of iron in the rim of the expanding lesion. And that's, of course, something we can translate into the message of iron rim-loaded microglia, which can be seen here on a 7 Tesla MRI machine. Although these are still research machines, but they bring us closer to in vivo pathology. And it's interesting to see that it almost reflects directly what in the past we could only see under the microscope. And that's really a great advantage because we can, in the future, clarify some of the questions we're still alluding to only on post-mortem material or on cases which were biopsied. And we also know these are not the regular cases, otherwise they wouldn't have gone for biopsy. But this is something where we learn about the evolution. There are even different stages, and we'll go through this, what they all mean in terms of our understanding on the advanced pathology of MS. So this is how we can see it in clearly described central vein sign, the appearance of the lymphocyte accumulation with inside the perivascular veins. And then the iron rim, which is expected to be shown on the aspect of a expanding active demyelinating lesion. And as you can see here, the accumulation goes into the perivascular space. And that's something which you can see in 90% of all MS lesions. And it's hardly seen in other inflammatory, metabolic, or vascular diseases of the CNS. So as it is confirmed with histopathology, it's also reflected now with in vivo pathology on 7T MRI. The interesting thing, though, the central vein sign is something which is already there since this first description by Eduard Rindfleisch in 1864. He said there isn't any MS case without this sign. So it's something we knew for some time, and we're coming closer to understand why is it there, why is it so typical, and how we can interfere already at this stage to have an impact on the progression of the disease. And I will focus on the first part of my talk about this central vein sign. So if you look at the accumulation as it is here with an overlaying disposition of the periventricular veins, which are the hallmark where the disease starts. This is what we know for years. What we have learned over the last 10 to 15 years or so is that there's also high accumulation of lesion in the cortical region, so close to the ventricular place, close to the meninges, and that comes into place rather soon. We have recently described other diseases which were in the past, all covered in variants of MS, but now have different connotation and disease entity called the MOGAD, so the MOG-associated demyelinating disease, where you see it's slightly different. It's more into the white matter, some are budding to the cortical region, but it's never clearly in this area around the ventricles where we typically see it in MS. And if you look at the staining of two important immune cells which are out there, you can see that there is this perivascular accumulation of lymphocytes. And you see in brown here the T cell description, and in blue the CD20 B cells. And as we can see, T cells you find here, but also in certain aspects in the parenchyma, the B cells almost exclusively stay in this perivascular room. And it's interesting to realize that because they are really in the front line to control what's coming from the blood into the brain, and probably have some association with the modification of antigens, which could induce the further pathological process in the CNS. And here it's just the combination of what are critical parameters. As I mentioned here, the B cells stay inside the perivascular space, the T cells can invade into the parenchyma, and it's classically a much larger perivascular vein lesion, and these are the ones which are much smaller, distributed across the white matter, as you can see it in MOGAD. So if we differentiate this and ask the question about potential target antigen in inflammation in MS versus MOGAD. For MOGAD, it's clear, it's the myelin oligodendroglicoprotein. That's why it has got its name. There are probably other CNS autogens for T cells while they transmigrate. This is still under debate. But the question is really out there, what's the antigen in multiple sclerosis driving the process? So the interesting thing, of course, is something which must be localized in the perivascular and meningeal tissue space, because that's the region where the immune system enters into the central nervous system. No specific CNS antigen, autoantigen has been defined yet, but potential candidates are always related to potential foreign antigens invading virus. And there is a hot topic, of course, since the latest publication Science about Ebv, Epstein-Barr virus, and I will talk about that. Some very interesting aspects about human endogenous retrovirus. And you probably don't know this, they really make up a large part of our genome, between 5 and 8% are encapsulated retrovirus, so they might have something to do, in particular HERV-W plays a role in certain aspects here. So the autoantigen in the perivascular or meningeal space is probably a B cell antigen, because that's where the B cells reside. That's where the B cells control what's coming from blood into the brain. So again, an infection of the B cells, an infected B cell, probably with EBV, could be really an important parameter to modify the autoantigens in a perivascular space, which is then driven by invading macrophages. So the concept of MS being an inflammatory disease is also rather old. It goes back to Pierre Marie, the successor of Charcot in Paris, and he claimed this in 1890, although it was a different infection there. He claimed it to be associated with typhoid fever. Now we are coming closer to probably a virus-driven process in multiple sclerosis. And it goes back to a heavily debated publication about the dysregulated Epstein-Barr virus infection in multiple sclerosis brains, published 2007, for the first time, where this was really described in a way that they are organized, almost looking at secondary follicle organs in the aspect of the meninges with the CD20 cells, and also an aspect related to receptors for chemokines, which attract cells getting into the central nervous system, and also having present there the deposits of immunoglobulin. And if you compare this now in the staining for the various antigens for Epstein-Barr virus, they are actually exactly co-localized with the CD20 cells. And if you look at the correlation of the presence of Epstein-Barr virus particles and CD20 cell count, both in the meninges and in the perivascular white matter, it's very highly correlated. And as you can see here, they're directly attacked by cytotoxic T cells, trying to suppress the process at this stage. And this seems to be deregulated, and thereby it can propagate into the central nervous system. That was a hypothesis that in the group of Francesca Loisi was pointed forward. And there was some difficulty to reproducing that, and this was still under debate whether this is really central or is it only an epiphenomenon. But it has definitely bring to our attention again the important role of B lymphocyte, not only as an effector cells producing antibodies, but also as a cell which regulates and activates T cells, as the possibility of producing and presenting antigen, and with their migratory role out of the primary lymph node to the germinal center, and then secondary germinal centers, which we find at the meninges, place them at the key stage of this important aspect of pathophysiology. And this is something you're probably reminded about what we have seen, particularly at the times when we started to seal the blood-brain barrier with anti-VLA4 antibodies. But at that time, there was all this told about the T cells. But now we know that also the B cell recruitment into the CNS, into the perivascular space, is very similar to what we have seen with T cells. Interestingly enough, there are some other than VLA4 and LFA1 important molecules which seem to be more associated with B cell transmigration, an antigen called ALCAM. And as you can imagine, there are already studies trying to block this specifically and thereby reducing the accumulation of perivascular B cells. You probably heard or even have read this study, which is just about a month ago, presented in January 23rd of this year, and was probably the most exciting first description of a new view on multiple sclerosis, or others would call a supporting view of the potential role of Epstein-Barr virus. So the longitudinal analysis reveals high prevalence of Epstein-Barr virus associated with multiple sclerosis. What have they done? I think the gratefulness to this tedious work, screening through over 10 million individuals, young individuals, mainly men, but also women with different origin, usually in the age around 20, which were recruited into the military service in the US, and they had compulsory sampling. That's what they do. And they have just looked at this in a way to follow at the different samples whether these were negative for antibodies against Epstein-Barr virus or if they seroconverted during that period of time. And they had made case controls of patients. These were overall 342 patients which developed MS over that period of time, and compared them to controls which didn't had the evolution of the disease at the same time point. So that's really comparable. And they were also compared into other parameters like gender, like their genetic background, and also age. And then they did different analysis, of course, the Epstein serology virus and similar virus, which does have a similar epidemiology, also affecting mainly at a younger age, cytomegalovirus. And then also the parameter, which seemed to be an important indicator, which we can measure in the blood already, the serum neurofilament light chains. Compare this to a virum analysis on a smaller population. It was seen that the age at first sample was very close, rather young, 16 years old. And then you see the age in which MS occurred in these patients. And we had different timeframes between the first sampling and the occurrence of multiple sclerosis. But what was astonishing, that there is a significantly higher number of patients, almost reaching 100%, as we have known, in the patient who developed during this observation period multiple sclerosis. And only what we know that the overall average of the seroprevalence for Epstein-Barr virus, 58 in the control group. But of course, the timely associated manner is very interesting. You see it in the majority already at the second seroconverted time. And here the baseline for the EBV positive in patients who had an increased hazard ratio to become multiple sclerosis positive, 38 times more likely than having not the disease. And this is the interesting control group. Nothing really of difference is shown in terms of the seroprevalence of a control virus, cytomegalovirus, and you can see it here in the uprising different serosamples. What was even more interest is how did the serum light chain, neurofilament light chain, behaved over time. And we know that this seemed to be a marker of neuronal destruction, so a neurodegenerative marker which we can measure associated with axonal damage in the periphery, in the serum. And it was seen that the patient who had the Epstein-Barr virus infection also had a significantly increase prior, most of them prior, some of them at the same time of the serum sampling of this light chain, neurofilament light chain. And again, in the controls which didn't develop MS, it stayed low. So there is a sequence now, and they wrote in their conclusion, and this is direct citation from the publication, the risk of MS increased 32-fold after infection with EBV, but was not seen with other viruses, including the similarly transmitted CMV. Serum levels of neurofilament light chain, a biomarker of neuroanatomical degeneration, increased only after EBV seroconversion. And then the final sentence in the conclusion, these findings cannot be explained by any known risk factor for multiple sclerosis and suggest EBV as a leading cause of multiple sclerosis. So a leading cause of multiple sclerosis as a conclusion of a highly cited, discussed paper published in Science, does it really tell us we need to vaccinate now our population at a younger age in order to prevent multiple sclerosis? We're not there yet. Maybe we will come to this. But first of all, let me explain a little bit more about what's really happening if EBV infects B lymphocyte. It activates the B lymphocyte. And EBV is a transformation of the B cells, and it was first described in B cell-derived tumors. We know it in Burkitt lymphoma, for example. And then it can induce both antibody production. It persists in these cells and can be activated in other stages. So it's a something dormant state And every time when the B cells encounter some activation signal, it might be reactivated as well and can propagate this way. It can induce the activation of T cells and, of course, the differentiation into plasma cells. So the question is, what is the hopeful or the major mechanism how Epstein-Barr virus induces all these probabilities and capabilities of B cells? And there have been in a very nicely recently published review on the role of B cells in CNS pathology, the different aspects how the B cells might behave. And it's interesting that in cellular communication, there has been described a new phenomenon, the exosome, which is an accumulation of certain proteins. And every time when there is not only cellular protein, but something foreign, for example, a virus is associated with this, they become aggressive and they can directly attack neurons and oligodendrocytes. We know about, of course, everything which is antibody mediated, but the fair amount of cytokines which are secreted, both in terms of pro-inflammatory and anti-inflammatory, puts the B cells into the stage of a regulatory cell and also a cell which can interact with T cells. So if something goes wrong in the B cell activation by a chronic infection, this is really a potential and very likely driver of the inflammatory aspects of multiple sclerosis, with the direct capability also to destroying central nervous system antigens and structures. So a recent publication in neurology has looked upon that in patients who had been isolated the exosomes from subject with MS, which express EBV-derived proteins, and they can activate monocytes into activated macrophages, and this can be translated also into activated microglia, and it's associated with activity of the disease. As you can see here, that the healthy control are in blue, the green are stable patients, and the activated patients, the active patients, sorry, had both aspects of highly expressed proteins like EBNA1 and LMP2A, which are both late activation proteins from the Epstein-Barr virus present in these exosomes. And now the next link between something which we know is important in multiple sclerosis is the genetic background. We know that there's a certain HLA phenotype which is more prevalent in MS patients than in controls. And we know that some of the potential modified exogenous risk factors, as they're listed here, again by a very interesting and still up-to-date publication from 2017, probably the highest association with the genetic background with a conduced odds ratio of 15 is with Epstein-Barr virus infection. So this rounds up the pattern that a certain genetic background with an Epstein-Barr virus infection can be a highly driven aspect of the disease later on. So we can derive a model now that asymptomatic or symptomatic infection with Epstein-Barr virus with a high viral load due to immune control and due to genetic background, maybe other cofactors involved in that as well, can induce B-cell proliferation, particularly in areas where B-cell follicle formation and EBV reactivation can occur, inducing an antiviral cytotoxic immune response, and by that, basically ignite the bomb that the inflammation goes astray beyond the perivascular space, invading then the activated T cells into the parenchyma. And the question is, with all of that here, EBV transformation, raised neurofilament levels, going to various different stages of the disease, asymptomatic, prodromal, CIS, multiple sclerosis, can we prevent MS with an EBV vaccine? And if you look at the accumulation of clinical trials data, there are already 2 in place looking at that in the very near future. So this is a domino model, you know this game, and we know that things can go like almost exponential once it's induced. And there are probably the genetic background, as I mentioned to you, the month of birth, the low vitamin D, which basically triggers the gun, but you pull it with the additional infection, which then can lead into full-blown MS. There are other risk factors, exogenous, which can be reduced, but there is the possibility at this stage already to intervene, and then hopefully will prevent the evolution of the disease. So this is something which is, not very old, but it has already immediately translated, or prior to this publication, probably with some knowledge before, that we will look into vaccination strategies to prevent MS in the near future. So I told about the central vein sign, and let me spend the remaining few minutes also now on this situation. What's this expanding lesion? What is in the area where we have activated microglia? Some of them, we know that these are lipid-loading macrophages carrying on the debris, but also propagating the disease into the central nervous system. And again, remember, exosomes from B lymphocytes infected with Epstein-Barr virus can activate macrophages, and that can induce neurodegeneration and secondary progressive MS. So we really have now the histopathological location where it starts, and we have a sequence of events triggered by the Epstein-Barr virus ending up in macrophages and microglia activated through this mechanism and then inducing neurodegeneration in secondary progressive MS. And we know that this has a direct impact not only on the degeneration, but functionally also interfering with what we need in order to maintain stability or regain function, for example, after an attack. This is synaptic plasticity. And that is highly impaired by these mediators, which are secreted by the activated microglia and macrophages. So we still have a chance by reducing this to readapt synaptic plasticity. And that's what we see probably in secondary progressive MS. If we see patients getting better, it's not a fixed deficit yet. It's probably in this area where we can then reactivate synaptic plasticity and regain function of the areas in the CNS which are not distorted. So highly interesting explanation of how we think drugs, which in the future we are probably giving more in patients with secondary progressive MS, are really able to interfere with this process. And if we summarize now the aspects which are the pathological subsets of progression, it all comes back to the initial part which I described to you, the cortical lesion and the diffuse inflammatory gray and white matter. And you still see the majority of the cells controlling this process in the perivascular cell space are B cells. Also in the cortical lesion, we know that the follicular dendritic cells or the follicular structures where B cells become activated, and also in the diffuse white matter, they play an important role in the control of the propagation of the destructive inflammation component, and thereby control also secondary antegrade and retrograde degeneration in gray and white matter, and finally leading into atrophy. As we're focusing more and more with clinical trials, phase 2 and phase 3, and some already approved in progressive forms of MS, I think there's a highly chance that with this intervention, where we still control the parameter at the late stage, which is associated with active inflammatory components of the disease, might be able to bring us closer to a life without multiple sclerosis. So let me summarize. Pathophysiology or pathology update 2022. I think the majority of the aspects which we have learned is we can describe aspects of the continuous process of inflammation and degeneration over decades with in vivo pathology now on the MS lesions, focusing on the central vein sign and the iron ring lesion, which are typical markers of in vivo pathology. And we know that the igniting factor comes closer to the important role of Epstein-Barr virus because it was shown in a young population, follow up over long periods of time with regular sampling, that there is an increase of up to 32-fold in MS patient associated with Epstein-Barr virus, followed by neurofilament light chain increase in the serum, and then clinically definite multiple sclerosis. EBV has multiple effects on B lymphocytes in MS. We have described that as well. And the induction of antigen-specific tolerance by mRNA vaccines associated with bystander immunosuppression is one potential aspect of the treatment in the future. And activated macrophages and microglia-induced neurodegeneration in the progressive phases of the multiple sclerosis. And clinical trial strategy focus more now on progressive forms and prevention of MS, like vaccination in the future. And with that, I would like to thank you for your attention and just showing you that it's almost up to become spring now. We had nice skiing holidays and working in a place which is so close to the first ski slope. You can imagine that's enjoyable working, and it keeps up your fresh memory at a slightly hypoxic level. Moderate hypoxia at 850 meters above normal also activates your brain. So you're invited to join me there for a session whenever you come across. Thank you very much for your attention.